Hey everybody, welcome back to the channel. I'm super excited today to be talking to you about a brand new series that I've been wanting to begin, but have just been kind of busy in real life. And I've finally gotten around to making the first presentation. And this particular series is going to be called Evolution in Light of Population Genetics. Now, there's many different ways to study evolution, and evolution has been presented in a myriad of different ways just here on YouTube. You can find tons of channels looking at the fossil record, looking at embryology, looking at kind of the basics of genetics, maybe giving you little snippets and some examples of how we can use genetics to understand the evolution of populations. But I feel like one of the things that's really lacking on YouTube in like an evolutionary biology context is looking at evolution from first principles, looking at it from the mathematical basis that was actually crucial in its formation, starting in the 1920s and onward. And so, what we're going to do here in this series is we are just going to hit the ground running and really tackle evolution in the light of population genetics. So, we're going to be explaining and examining evolutionary theory from first principles. And why does this matter? Why is it so important that we think about evolution in this context? One of the big questions in the life sciences, in evolutionary biology, this was actually featured in one of the top Science magazine released a top 100 unsolved mysteries in biology issue a while back. And this was one of the questions that they had. So, why are genome sizes so variable across the tree of life? What I've showed you here is just a figure of archaea, bacteria, protist, all the way up to mammals, and you can see huge variation in the genome size. Note that this is a log scale down here, so this is 1 megabase, and this is 100,000 megabases, right? So there's a huge range of genome sizes across the tree of life. Here are humans right here, kind of a middling genome size. Um, so obviously this leads to the next question. Why is there virtually no correlation between genome size and organismal complexity? And why are eukaryotic genomes riddled with parasitic DNA while prokaryotic genomes are largely free of it? So these are some questions that, without thinking about the population genetic environment in which these organisms revolve within, you're going to struggle to understand these questions of genomic architecture. Why do they look the way that they do? Why have they evolved the way that they've evolved? And so we're going to revisit this question later on down the line after we've kind of laid the groundworks of some population genetic intuition. And then we're going to come back and see if we can apply that intuition to some of these kinds of questions, as well as a range of other questions that I think could be interesting. I'm going to present some quotes here from two famous evolutionary biologists. The first one is Sewell Wright. Sewell Wright is one of the so-called trinity of the modern synthesis. Sewell Wright, along with R.A. Fisher and J.B.S. Haldane. And in 1930, in his book, Evolution in Mendelian Populations, he states that the present status of genetics is that any theory of evolution must be based on the properties of Mendelian factors, and beyond this, must be concerned largely with the statistical situation in the species. And this kind of gets at the root of what evolution is. What's the purpose of evolution? How do we apply it in a theoretical framework? And I think today, even almost 100 years later, we still follow Sewell Wright's advice here, and that evolution is largely concerned with the statistical situation within the species. And a more concise and pithy way to say this was in Michael Lynch's 2007 book, Origins of Genome Architecture, in which he states that nothing in evolution makes sense except in light of population genetics. And this is a pretty bold claim. You might recognize this as a rephrasing of Dobzhansky's maxim that nothing in biology makes sense except in light of evolution. Lynch has taken this and extended it a little bit further. That a lot of these big questions in evolutionary biology, such as the origins of genome architecture, why genomes look the way that they do, don't make any sense if you don't consider the population genetic environment in which these organisms actually lived. Okay, so what are some of the broad overarching goals of this series? So number one is defining evolution as a mathematical process of populations. So spoiler alert, there's going to be some math. Don't be afraid. I'm going to walk you through it very easily. We're not going to do like any super in-depth things. but there will be some math. You will see some math on the slides. Don't let that scare you off. Second, we're going to examine each of the four mechanisms of evolution in their mathematical context. Then we're going to introduce some more complex population models. We'll start off with very simple models, and then we'll just kind of build on those. And then finally, one of the things I'm really hoping to get across from this series is to demonstrate the dependency on macroevolutionary patterns on population level processes, right? So things happening at the individual population level will then echo across the tree of life, right? It leaves traces, it leaves its signature on broad macroevolutionary patterns. And so oftentimes in biology, we study microprocesses, macroprocesses, and then we forget how these things are linked together, right? And so one of the major areas of my research is to actually look at how these two processes link together. And so that's the thing that further on down the line, when we introduce some of these models, I hope to revisit this and see if I can show you how these processes are actually linked. And then one last thing before we dive into this part one segment is this thought question. And this is almost a philosophical question. It's a question that dates back long before Darwin, all the way back to like Aristotle and the Greek philosophers, is what is the nature of a population, right? So you can think about the dashed line in each of these two boxes as the trajectory of the population. And in the left, you can see the trajectory or the ultimate form, the normal phenotype is stagnant. And then you see some morphological change through time, but it's just variations on a theme, right? It doesn't actually go in any direction. It doesn't change dramatically. You're just looking at noise around a theme, right? And this is really gets back to like Plato's idea of the form, where you have some like perfect form, and then all of nature is just a variation on that perfection, right? And so you never actually see a change in species. Before Darwin, this was the way that we thought about populations, right? We did not think about them as changeable entities. So species were immutable. There were some perfect form, and any kind of variation you saw were just aberrations, right? Alternatively is the Darwinian view, or the evolutionary view, that populations change through time, right? That there's variance around that change, but that you can see a gradual change in either morphology, gene frequencies, allele frequencies, et cetera, that things change. And this shows it like it's directional. It's not directional in any sense. Evolution is absolutely not that. But I'm just showing it to kind of counter the sort of stasis that might be expected under, like a model that all species are immutable, right? So just kind of think about this, keep this in the back of your head, that before Darwin, the figure on the left is how we thought about populations, right? So one of Darwin's biggest contributions is overturning that view that populations could change through time. Okay, so part one, Hardy-Weinberg equilibrium, what I'm subtitling The Reluctant Intruder, and we'll see why in just a second. So many of you, if you've taken even basic college biology, you know Hardy-Weinberg. This is like the absolute basic population genetic framework. Every biology student learns it. I don't know if they teach it in the non-majors, but maybe they do. You should be familiar with this concept. But one of the things that I want to do here, and what I'm going to do across this series, is not only present you just like, here's the model, yay, but kind of give you the historical context, because there's some interesting history here, especially with this model. So I'm going to take something that may have been incredibly boring in your biology intro bio course and try to make it cool by throwing some history at it. Okay, so bear with me. As we kind of go through this. So first and foremost, I'm not going to spend a lot of time on this, but I just need to lay this out there so that we have the understanding that it's here, and that's Mendelian genetics. And there's three key things that we need to know about Mendelian genetics, and these are his major fundamental discoveries. One is that inheritance is particulate, right? He overturned the predominant idea that inheritance was blending. This is actually treated on a mathematical level in R.A. Fisher's book, The Genetical Theory of Natural Selection. If you check that book out, it's like the fundamental foundation of mathematical evolution. Check it out. He treats the blending theory of evolution and the particulate theory of inheritance, I should say. And he shows that when inheritance is particulate, natural selection is very, very efficient. When it's blending, it's not. So this is why Mendelian genetics was super important for a discovery for forwarding Darwinian natural selection. Secondly is the law of segregation. This is that gametes receive only one copy from their parents, and that which copy they receive is randomly drawn from the parent. So if we look up here, this is the parental generation, where you have your white flower, your purple flower. For the purple flower, they have two alleles, a big P and a big P. So they're homozygous for the big P. All right. The parental generation is only going to get one of those P's from that parent. That's the law of segregation. And which P they get is random. Okay. Finally is the law of independent assortment. And this is that genes are inherited independently from one another. So you can imagine if you have a purple flower that's tall, right? So you've got purple and tall. These are two discrete traits, right? So the gene for purple and the gene for tall are inherited independently of one another, right? They're not linked, okay? We know examples of genes that are linked. This process is called linkage disequilibrium, and we will talk about that down the road. But for now, and especially for Mendel's pea plants, a lot of the genes that he looked at were independently assorting. So those are the three fundamental discoveries of Mendel. Also to note is I've just kind of given you a kind of primary example of the sort of crosses that he did. So these are self-cross. These are homozygous crosses where you have a white flower is homozygous for recessive P. The purple is homozygous for the dominant P. The F1 generation is all heterozygous because of that. And then you get the reemergence of the traits in the second, in the F2 or the second filial generation. And then you can see you have one dominant, one recessive, and then you get two heterozygous. So it's a 1-to-1 or a 3-1 morphological or genotypic ratio. Okay, so before we move on to this, what I would like to do is give you a little bit of background. I'm going to actually read to you just a little bit of an excerpt from a paper by A.W. Edwards. And this particular paper is super interesting to me because he goes through the history of the Hardy-Weinberg principle, okay? And what's curious about this is that Mendelism was still being discovered. It had just been rediscovered in 1900. There was a lot of debate about whether or not, one, it was correct, how applicable it was, and whether or not it worked well as a theory of evolution, okay? So what this perspective piece did is what Edwards did is he basically presented the history of G.H. Hardy, who is the Hardy and Hardy-Weinberg, talked a little bit about Wilhelm Weinberg, who also contributed to this notion, to this theory, but then talked about it a little bit, kind of people that were very close, right? And this is an important point in science, is that so many people get very, very close to the right answer, but don't quite get there. And this is kind of one of those sorts of stories. So he writes it on January 13th, 1908, which is actually before G.H. Hardy wrote his letter to the journal Science. Wilhelm Weinberg read to an evening meeting in Stuttgart, Germany, a paper in which he, quote, described the general equilibrium principle for a single locus with two alleles. And then he goes on to give the famous p squared plus 2pq plus q squared equation, right? The binomial equation that you can get from multiplication tables. And he writes that this situation appears much different when Mendelian inheritance is viewed under the influence of panmixia. He's referencing Mendel is looking at his proportions in selfing, whereas Wilhelm is presenting his proportions in panmixia. And we'll get to why that's important in just a second. So Wilhelm, starting with arbitrary proportions, little m and big M, you can think of this as little p and little q, of each of the two homozygotes, he obtained by, quote, application of the symbolism of the binomial theorem, the daughter generation, p squared plus 2pq plus q squared, right? And this is the exact same distribution as he got in the previous one. So another generation of random mating, Edwards writes, led by direct calculation to the same proportion among the offspring, and, quote, thus we thus obtain the same distribution of pure types and hybrids for each generation under Penn-Mixia. So that is Hardy-Weinberg, right? That is the central discovery of Hardy-Weinberg, that if you have random mating, such that every allele is drawn at random from the parental population to produce gametes, and then the distribution of those genotypes in the next generation exactly match those in the parental, right? That's the fundamental discovery. So he begins the paper just by kind of giving Wilhelm sort of a shout out, yeah, like, yo, bro, yeah, we know you were first. Here's a little bit about you. However, in Britain, like, this paper wasn't recognized in Britain until like several years later. So Hardy, his name gets to go first because of this. And Hardy's story is a little bit more interesting. So one of the things to note is that there was a disagreement among the Mendelians and what were called the biometricians. Okay, and these are people that were looking at continuous variations in traits, and they were trying to come up with mathematical and statistical scaling laws for like what kind of traits were inherited and the nature of inheritance based on continuous trait variation. And some of the biggest debates at this time were between people like Udny Yule. These are some names you might know: Yule, Pearson, Punnett. These are guys that like are fundamental in the foundation of a lot of statistical measures, right? Like Pearson's correlation coefficient. Yule introduced the normal distribution. Punnett is famous for the Punnett square, right? Like all of these names should be somewhat familiar to you. They hated each other. So they were like at each other's throats in the early 1900s about what is the mechanism of inheritance? What do you expect to see? It was pretty acrimonious, this particular debate. There's some great papers on this. If you're interested in the history, you should check it out. Okay, but basically the way the story works is that Punnett went to a mathematician named G.H. Hardy. And Hardy is famous for doing all kinds of really cool things in like pure mathematics. He was not a geneticist, not a biologist at all, just worked in pure mathematics. And Punnett was having a disagreement with Yule and Pearson and some of the other biometricians. And so he came to Hardy to see if Hardy could help him out. It's like, bro, they threw something at me. It's a mathematical question. I don't have the mathematical chops for it. Can you help me out with this? Right? So roughly speaking, Punnett's concern, and this is Edwards' writing, Punnett's concern was why in a random mating population, the dominants did not in the course of time drive out the recessives. Okay. And so this was the question that Yule put to Hardy. So what I'm going to do is I'm going to advance the slide. Here we go. So this is Yule, and at a meeting, he gave an example of brachydactyly, and brachydactyly is short fingers or end toes. It's like short digits, and it is caused by a dominant allele in humans, and we're going to call that dominant allele the big B. Normal fingers, however, we'll call little b, and it's recessive. And so Yule at this meeting calls out Punnett, and he says, okay, if Mendelism is correct, if Mendelian inheritance really is the way that that alleles are inherited, then brachydactyly should be present, or will eventually be present in the human population at a 3-to-1 distribution if Mendel was correct. So let's think about where he kind of gets this intuition. So again, all I've done is I've replaced flowers with normal hands and brachydactylous hands. And then you can imagine homozygous mating with a homozygous, you get a full heterozygous next generation, and then you get a distribution of dominant, 2 heterozygotes, and the homozygous recessive for the normal hand, right? So as you can see, this is a 1-2-1 where it's homozygous, 2 heterozygous, 1 homozygous, or a 3-1 because 3 of these individuals are brachydactylous to 1 that are normal, right? So to him, he thought, okay, well, why is it then, if Mendel is right and this is the form of inheritance, why don't most people have brachydactyly, right? Why isn't that something that most of the population suffers from? So as we go forward, I want you to take a minute and think about this and see if you can figure out what's wrong with his calculation, okay? All right, so this is Hardy's response, all right? Hardy took it very quickly, worked out the math, and then he wrote it up, sent it into the journal Science. And this is a letter to the editor, and it's titled Mendelian Proportions in a Mixed Population. And he writes with great shade, I am reluctant to intrude in a discussion concerning matters of which I have no expert knowledge, and I should have expected the very simple point which I wish to make to have been familiar to biologists. However, some remarks of Mr. Udny Yule, to which Mr. R.C. Punnett has called my attention, suggest that it may still be worth making. And he then goes on to lay out the mathematics of what would become the Hardy-Weinberg principle. Now, while this makes Yule seem foolish, and Yule and a few of the others seem foolish, I do want to note that a lot of these guys were really close to the right answer and they just didn't know it. So I want to read you an excerpt. Again, this is from the Edwards paper, and this is from Yule's paper. So Yule actually wrote a paper where he was considering dominance, recessive, Mendelian proportions, etc. And he writes this. The first question to be asked in such a discussion is that does not seem to have occurred to any Mendelian followers. What exactly happens if two races, big A and little a, are left to themselves to intercross freely as if they were one? Now, when homozygous A's, and big A's and little a's, are first intercrossed, we get a series of uniform hybrids. Remember, this is the F1. We get a series of uniform hybrids. When these are interbred, we get the series of three dominant forms, two hybrids and one pure. Remember, this is the 1-2-1 or the 3-1. And one pure. If these are again intercrossed at random, the situation remains unaltered. Dominant and recessive gametes are equally frequent, and consequently conjugation of a dominant gamete will take place with a recessive gamete as frequently as with another dominant. He wrote this in 1902. Okay, so again, he's right there. He's on the cusp of getting this. So there's a kind of a curiosity, and Edwards points this out a little bit in his paper, that Yule is so close to the right answer, and yet he appears to not understand it, right? He poses this question to Punnett about brachydactyly, even though he knows what the right answer should be. So again, this is kind of an interesting point of somebody that's very, very close, but not quite there. Pearson, another biometrician, is also very close, and he writes in one of his papers. Actually, this might have been in one of his books, but he writes, "It is thus clear that the apparent want of stability in a Mendelian population, the continued segregation and ultimate disappearance of the heterozygotes, is solely a result of self-fertilization. With random cross-fertilization, there is no disappearance of any class whatever in the offspring of the hybrids, but each class continues to be reproduced in the same proportions." So again, these are two guys writing before Hardy and Weinberg that are right on the cusp of the right answer. They are so close. They don't believe in Mendelian inheritance, I should point out. These two people were like antagonistic towards Mendelian inheritance, and yet they're so close to the Hardy-Weinberg principle. Okay, so what we're going to do, let's now look at Hardy's model. What did he actually put out there? What was the math that he derived? And let's see if we can walk through it together. Okay, so very, very important. Hardy makes a series of assumptions, and Hardy is very good in his paper about laying out these assumptions because he's a mathematician, right? And mathematicians are very good at saying, assuming X, Y, and Z, then we can have A, right? Assumptions. 1, infinite population size. This is critically important because if you don't have an infinite population size, then what you get is sampling bias, right? By random chance, you will get fluctuations in frequencies just by chance alone, just by sampling error, right? So we're assuming we can sample the population perfectly such that our sample exactly matches the proportion in the previous generation. That's the first assumption. Second, no gene flow. So there's no other populations that are throwing alleles into our little hypothetical population. No mutation. You should note that those two things do basically the same thing, right? Mutation introduces new variation. Gene flow brings in new variation. Both of those things promote variation. Whereas the first one, infinite population size, removes variation. No selection, obviously, no natural selection because that would be changing allele frequencies. Random mating. This means both no sexual selection, but this also means no spatial structure. Okay. This is often only talked about in the context of sexual selection, but really what they mean when they say random mating is panmixia. And what panmixia means is that everybody chooses their mate at random from a gene pool. Whereas in reality, a lot of species choose their mates by who's nearby, right? So what you end up with is a kind of spatial structure where nearby individuals share more alleles in common than more distant individuals. That's what we mean by random mating, right? That individuals, no matter how far away they are, choose their mates at random. And then finally, equal sex ratios. If you've got more males, more females, then you can skew those frequencies just by the fact that one sex is more dominant than the other. Okay, so with those assumptions in mind, let's lay out our model. And I just made up some numbers for this. So these are not the actual proportions of brachydactyly in human populations, okay? I just picked these numbers because the math is very simple and straightforward. So let's say 98% of the population has the normal phenotype, and 2% are brachydactylous. Obviously, that is not the real number. It's way, way more skewed than that. But let's say that's the case. And let's say then you could sequence the whole population. You know exactly what proportion of them were homozygous for normal, heterozygous, and homozygous for brachydactyly. And this is the distributions we got. So obviously, since this is the recessive phenotype, you know that the number that you see is exactly equal to the proportion, right? So those two match. But since this is the dominant one, some portion of those are heterozygous and some portion are homozygous. So the heterozygous is 0.02, and then the homozygous is 0.0001. So very few homozygous, a lot more heterozygotes, and then most people are normal. Okay, what's the frequency of those particular alleles? All right. Oftentimes, we're interested not just in the genotypic frequency, which is what I just showed you, but the allele frequency. What's the frequency of those specific alleles in the population? So the frequency of the little allele, B, equals to the frequency of the homozygous genotype, which is this one here, plus half the frequency of the heterozygous, because half of the alleles in a heterozygous individual are the little b, right? So you take 0.98 from here plus half of 0.02 from here, and that gives us the frequency of little b as 0.99. For those of you sticklers, there's some rounding here. Don't lose your mind. I'm just keeping the numbers simple and like doing some rounding. So yeah, 0.99 is what we're gonna roll with. All right. So then what's the frequency of the big B? We do the exact same thing. We can take our homozygous individual for big B, which is this number here. We plug it in, plus half of the heterozygotes gives us 0.01. All right, so the frequency of the little b is 0.99. The frequency of the big B is 0.01. We can obviously have just intuited that from the simple relationship between the big B and the little b. Note that the big B frequency is always 1 minus the frequency of the little b, and vice versa. Right? The frequency of the little b is 1 minus the frequency of the big B because they're dependent on each other. And we're assuming it's a biallelic system, I should say. So we can see that we could have just plugged in the frequency of the little b, which is 0.99, and gotten the correct answer in the end, just to kind of show you that relationship. All right, so I've put that number up here. All right, so now we have our allele frequencies, and then we have our genotype frequencies. All right, so what will the trait distributions be in the next generation? And this is the thing. This is the critical thing that Pearson and Yule are right on the cusp of, but that it took Hardy and Weinberg to actually put into mathematical formula. So what we're going to do is what Hardy calls a simple test or a simple multiplication table. So what we're going to do is we take all the possible combinations of alleles, because remember, if there's random mating and we can get exactly the same proportion of genotypes from the first generation into the second, then all we need to do is make a complete contingency table of all possible pairs, right? So if you've got the big B matches with a big B, then you get B squared, right? That's the frequency that you should see that happen. For the little b and the big B, since those two things are different, we multiply them together as big B, little b. We do the same thing down here, little b, big B, and then b squared. So we've just multiplied each one of them across that table. And what you should notice is that we're getting pretty close to what you'll recognize as the Hardy-Weinberg, right? So let's then stretch these out. We're going to plug the numbers in. So big B, big B. So that is just 0.01 squared, because remember, big B is 0.01. For little b, big B, it's 0.01 times 0.99. Same thing down here, 0.01 times 0.99. And then we get big B, or little b squared for 0.99 squared, okay? So what's the frequency of individuals that are homozygous for brachydactyly? Again, that's B squared. And then the number of, and then the frequency of that genotype should be 0.0001. As you can see already, and what you already know is going to be true, is that that is exactly the frequency that it was in the previous generation, right? Second, big B, little b, this is for the frequency of the heterozygotes. Since we have two big B, little b's, we put them together, and that gives us 0.02, okay? And again, that's the frequency of the heterozygotes in the previous generation. And then lastly, we have 0.98 for b, little b squared, and that's the frequency of the normal genotype. And you can note very easily, we put all of those numbers together, we get b squared plus 2bq plus 2b little b plus b squared. So yeah, it's like goes straight to p's and q's. But as you can see, we've just derived Hardy-Weinberg, right? Super simple, like super simple math, super simple multiplication table. And this is why it's so amazing that people were struggling to get to this point. We're right on the cusp of it, and yet just couldn't quite get there, right? Okay, so what's the problem with this calculation? Why did he get this wrong? Why did he make that? Why did he punt this question? If he, for one, seemed like he knew what the answer should be, and yet couldn't seem to figure this one out. And we just did it very simply, and we are nowhere near as smart as a Neil could possibly be, okay? Well, he assumed allele frequencies were 0.5. That's why he got this wrong. And you can see this very clearly if you were to plug in allele frequencies that are 1/2, right? So if you take p squared plus 2bq plus q squared, as I've been trying to say this whole time instead of b's and little b's, you take 0.5 squared plus 2 times 0.5 plus 0.5 squared. That gives you a 1-to-1 ratio or a 3-to-1. So the only time that you're going to see that exact phenotypic ratio is if you assume that those alleles are present in 1/2. All right. And this is what Edwards in his paper goes on to talk about ad nauseum. It's like how these guys, or for like what possible reason it was that they constantly assumed a gene frequency of 1/2. The explanation that Edwards seems to give is that Mendel, when he was working with his pea plants, started with crosses, and so you knew the proportions were 1/2 and 1/2, right? Because that's just where he started. And he's only tracking one lineage down, and that's the population, right? So when you start as self crosses, the frequencies of those alleles in the self crossed is exactly 1/2 and 1/2. So I think the problem that a lot of the biometricians were having in accepting Mendelian genetics is they were getting caught up on this idea that they had to be 1/2 and 1/2, right? So that was Yule's problem, and probably Pearson's problem as well. Okay, some final thoughts. Let's wrap this all up, and hopefully you learned something interesting, at least historically. I'm sure, again, most of you probably knew about Hardy-Weinberg, where it comes from, how it's derived. But the history behind it, I think, is pretty neat. So hopefully you learned something there. So how useful is the Hardy-Weinberg equilibrium as a null model for evolution? This is what it's often pitched as. And you can think of it as a model of no change, right? It's a model that assumes that there is allele frequencies are going to be constant through generations. So how useful is it? Well, for one is that this model is always violated, right? It's always violated in real systems because of, if none of the other reasons, because there is no population that's infinite, right? All of those other things can sometimes be true, but no population on the planet is infinite in size. So that one thing alone always means that the model is not going to match perfectly. However, it can be close. So if you've got a really large population that's mostly following generation to, and you're just following it on a couple of generations, it can approximate Hardy-Weinberg pretty reasonably. And in fact, if it deviates in one generation, one more subsequent generation of random mating, no selection, etc., puts it right back in Hardy-Weinberg, right? So even if there's selection in one generation, changes it, then they go back to random mating, they're right back in equilibrium. So it is actually a very useful tool for population genetics. It's a useful model for beginning. And what we're going to basically do is we're going to take this model in the subsequent videos and we're going to expand it. We're going to expand it to include selection, mutation, migration, drift. We're going to look at assortative mating and all of those sorts of things. And what you're going to find is that basic equation is at the root of all of those things, right? So that's why it's a super important model. And then I've already kind of answered this, is which of the assumptions are likely to be the most violated? The infinite population size is always violated, right? It's violated more than any of the other ones, because there is never a situation in which a biological system has an infinite population size, right? Okay, so that's what I've got for you today. Again, hope that you found this interesting. Hope that you learned a little bit of history about some of the acrimonious disputes in early population genetics. I think that the Hardy dispute is pretty interesting, that this pure mathematician got dragged into a debate among biologists. It's not the first time that's happened, and it probably won't be the last time that has happened. And yeah, so if you found this interesting, hit a like, leave me a comment, something that you're thinking about, something that maybe wasn't clear to you. If I wasn't clear on a certain concept, let me know. I'd be happy to chat about it in the comment section. And again, thanks so much for being here, and I'll catch you next time. Thanks.